Hello, everyone. I'm here to discuss a very important technique in molecular biology. It is called polymerase chain reaction technique. What is polymerase chain reaction technique? It's a simple and easy to perform technique, which is also very rapid, using in vitro amplification of a target gene. That is, several copies of a short DNA fragment can be made. The template DNA is required from which target sequences can be copied. A master mix with primers, that is 2 short oligonucleotide sequences, and a heat-stable DNA polymerase are also required. There are several steps in polymerase chain reaction technique. The first step is known as the denaturation step. It is the separation of the DNA double helix into 2 single strands upon the application of heat at 95 degrees Celsius for 1 to 2 minutes, followed by the second step, which is known as annealing. This involves the binding of primers to specific complementary nucleotide sequences on the DNA template at a lower temperature of 55 degrees Celsius for 30 seconds to 1 minute. The third and final step in the polymerase chain reaction technique is extension. This step involves the addition of more nucleotides to the annealed primer end by the DNA polymerase at an increased temperature of 72 degrees Celsius for 1 to 2 minutes. These steps are carried out in a thermocycler. Between 25 to 35 cycles are required to produce million copies of the targeted gene. Figure 1 shows the steps in the polymerase chain reaction protocol. The first step is the denaturation of the DNA helix, separating the double helix into 2 single strands, followed closely by annealing. That is, the 2 primers are added in a 5 to 3 prime direction. And this step is influenced by the GC content of primers. The third step is the extension. This is where Taq polymerase aids in synthesizing DNA at a temperature of 72 degrees Celsius. Figure 2 shows the gel electrophoresis image of MEK aging. This is just an example of how PCR products are visualized. Here is the point of application and the size marker ladder. MEK aging is found in methicillin-resistant Staphylococcus aureus, and the amplicon can be visualized in the gel electrophoresis at a band base pair size of 533, which is represented here. What are the applications of polymerase chain reaction technique? It allows in virology, it allows the detection and characterization of viral nucleic acids through the amplification of specific regions of interest. So genotypes, variations, subtypes, mutations, and standards of genotypic resistance of these viruses can be determined. However, it should be noted that reverse transcription PCR, RT-PCR, is used, which facilitates the detection and application of PCR products obtained from viral genome in the study of viruses which cause infectious diseases. Also in mycology, PCR or polymerase chain reaction technique can be used to detect trace amounts of fungal DNA from environmental samples. In patients with neutropenia caused by aspergillus infections, they can be detected using PCR technology. The disease caused by this fungus is very difficult to diagnose due to the poor sensitivity of the culture medium. In parasitology, diagnosis of parasitic infections and disease can be enhanced using polymerase chain reaction technique. Many parasites are not culturable in the laboratories. Also, conventional PCR has been used for the identification of microbial pathogens in clinical microbiology laboratory research. Diagnosis of bacterial infections and diseases can be done by PCR technique. Other applications of polymerase chain reactions include its use in forensic science, also to detect genetic diseases, DNA fingerprinting, analysis of phylogenetic relationship is used in mapping genes and sequencing genomes. Here are some selected references for further reading. Thank you for listening.